Welcome, everybody. So this symposium is organized by AMCRA, by Sciensano, by the Federal Agency for the Safety of the Food Chain, the Federal Agency for Medicines and Health Products, and the Federal Public Health, Food Chain Safety and Environment of Belgium. And this year, it's also our honor and pleasure to announce that the symposium is organized under the auspices of the Belgian presidency of the Council of the European Union. So welcome everybody who is physically present here, but we also have a lot, actually much more than the number of participants that are physically present. There are also a lot of people that attend the symposium via webinar. So it will also be recorded by by video, and the video will be placed afterwards on the website of the AMCRA. You have seen that there are also some posters outside, and you can view them during the coffee break or the lunch. And for the people that attend digitally, you can also see the posters via the website of the conference or via the QR code that is visible here on this slide. We have a keynote presentation and a few short selected oral presentations in the morning, and we will have the questions after each presentation. This morning there will be no translation. In the afternoon there will be simultaneous translation. So I'm happy to introduce our invited keynote speaker, and this is Jean-Yves Madec. Jean-Yves is doctor in veterinary medicine, PhD in public health, a postgraduate on antimicrobial resistance in human medicine. He is a molecular microbiologist and a researcher at ANSES in France. He is the scientific director of the antimicrobial resistance topic at ANSES and head of unit at ANSES Lyon. His research interests focus on molecular genomics and epidemiology of antimicrobial resistance in a One Health perspective, and he is an active participant and leader in European and transnational research projects. He is an active contributor to the Resapath network monitoring antimicrobial resistance in clinical animals in France. And he provides senior level guidance, expertise and support to the implementation of the One Health National Action Plan on antimicrobial resistance and antimicrobial use in France, notably in the agri-food sector. We are very happy to have you here, Jean-Yves, and we are looking forward to your presentation, which is entitled How to Monitor Antimicrobial Resistance in Diseased Animals, the example of the successful, may I say, French Resapath network. The floor is yours.
Okay, so hi everyone. Thank you very much for this very kind introduction. I'm very happy to be here. Thank you for inviting me. Happy because it's a very important event on EMR here in Belgium. Also happy because I can meet many colleagues, even those I have never met before, except by Teams, Zoom or whatever. So it's, yeah, thank you very much. And special thanks to Fabiana and Damien. I don't know if he's here for now, but thank you very much. Yeah. So the purpose of my talk is a brief introduction, and I'm also honored to open this event. Brief introduction on how we perform EMR monitoring in diseased animals in France. So I will try to convince you that, and also maybe convince the public authorities that it's highly important to monitor EMR in diseased animals. And just to stress a bit on EMR surveillance in Europe altogether, I mean humans and animals together, just to note that in human medicine, EMR monitoring is based on diseased patients from data originate from hospitals, from clinics, from the community, and that's it. And that's highly, highly useful for national action plans. And here is an example here where you can correlate what happens in a specific infections here for pneumococci in children and resistance to penicillin of pneumococci and the national action plan here, a measure taken by national authorities like vaccination. So it's highly correlated. But in veterinary medicine, it's quite the opposite. Most data originate, at least at the European level, from healthy animals. And the reason for that is because the angle of the EU is food safety. And the EU sovereign system on AMR in animals is dedicated to the protection of consumers, to protect consumers from AMR. It's coordinated by EFSA, it's under EU legislation, it's coordinated, the job is done by is coordinated by the European reference lab, the local job is done by the national reference labs. Everything is collected in healthy animals, poultry, pigs, and cattle, but healthy at slaughterhouse, and meat, specific methodology. So it's highly helpful. It's not to say that it's useless. It's just to say that it gives some information, but not all the information. It provides valuable insights on AMR in animals, but that, in our view, not sufficient. And the limitations, again, is just to say that you need complementary systems. The limitation is that as soon as we are talking about national action plans, when you urge vets to do something, to reduce the use of antibiotics, you need AMR data in the specific infections they are dealing with. If you give AMR rates in healthy cattle to a vet dealing with diarrhea every day in calves, it's not sufficient for him. He needs to have AMR data close to the daily job. So that's the first reason. And also the second reason is that you don't consider cats, dogs, horses, and many other food animal species. You don't consider in this type of surveillance certain important phenotypes like MRSA. MRSA has been the topic of a baseline study, and will be another one. But it's not a routine, because MRSA is not a food pathogen. So you consider Salmonella, Campylobacter, commensals, but MRSA is not included, animal pathogens. So you need to collect AMR data from diseased animals, from infections. And another argument is that it doesn't make the One Health easy if you collect data in healthy animals and to compare them with data from diseased patients, because they are completely different contexts. It's easier to compare data from, for instance, urinary tract infections in animals and in humans. That makes more sense. So diseased animals, it's a major gap at EU level. And this is what we think it would be important to set up in Europe. And this is what's just the example of the French system. The Resapath system was founded by my former boss, Jean-Louis Martel, is a vet, is retired now, and I took over his role, but he's the guy with the brilliant idea to start in 1982, where AMR was not a topic, at least at political level and so on, because he had some friends and took some beers and said, okay, those friends were heading laboratories, peripheral laboratories, and they decided to put the data together and to see what happens, to see if there were trends or, well. So he was the founder, and yes, it's a long-term system. You can see here, the 14th birthday 2 years ago. You can have access to the data online. This is something we have developed here. You just have to choose the animal species, bacteria, antibiotic, infection, type of infection, and everyone, it's public, but the vets, the farmers, everyone can go to the raw data. We can produce data with different angles. This is an example of the geographical distribution of the proportion of resistance in a specific infection, in E. coli, with regard to different antibiotics. And here it is whether the districts are above or below the national figure. And this system was evaluated by a method you may know, OASIS method, with good figures as well. I will not go into some details of the sections, but this is something which is important to evaluate your system, the performance of your system. So, how does it work? It is voluntary. It means that nobody is obliged to do that. So when a system is voluntary, you need to think of a win-win system. So there is an agreement between ANSES and the peripheral labs, which are the ones performing antibiograms for vets in their daily life. And we told them, your data interests us. We need your data to produce trends, to know what happens in the different infections, different bacteria and so on. And we will help you, we will provide expertise to you, we will help you to perform good, correct antibiograms. So the benefit for them is that they can say, okay, to their clients, to the vets, we are good laboratories because we perform antibiograms in the correct way. And we are supervised by ANSES, who are experts in AMR, blah, 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 blah. And so our antibiograms are of good quality. So that's highly important for them. This is a win-win system. And we collect data and we produce trends. So the objectives of Resapath are these three mainly. First, to monitor AMR. This is our objective. We need data to produce trends per infection, per bacteria, antibiotic, and animal species. Okay, and this is a report we produce each year. It's free also on the internet, in French, English version. And this is highly helpful for the authorities because they can say, okay, where are the hotspot of AMR? Okay, in cattle it's not mastitis, it's rather diarrhea in calves. Okay, we need to put efforts on this subsection in cattle. The second objective is to collect isolates and to do some molecular work and to see what are the different clones or different plasmids or different type of AMR is circulating in animals, whether it's similar to humans or not. And I will insist on that, we provide technical support to local laboratories. That is highly important. If you miss this, it will not work because they need that. They don't care about trends for the national authorities. They are local actors. They need something for them. So to monitor AMR, I will not go into much detail because I will not conscious of time, but just to highlight a bit, everything is online. We have the presentation as well. So we produce a report and trends. This is an example of trend. This is the nice decrease of resistance to fluoroquinolones in E. coli in different animal species. You can see the decreasing trend. This is an old figure. You can see that it's stopped on 2016. This is at the beginning. The National Action Plan started in 2012. So you can see 1 year or 2 years after the start of decrease, the resistance to ceftiofur in E. coli in calves. So it's highly important for authorities to say, oh, I put some measures in place, it works. Resistance is decreasing. That is very important. And we had in France a very, very important decrease in animal exposure to antibiotics in France. These are the figures produced by the national agency within ANSES in charge of the monitoring of the use of antibiotics. So a very marked decrease in critically important antibiotics. So we produce trend. This is our job. This is for the ministry, for everyone, for the human medicine to say, okay, we reduce antibiotics or put some measures in place, and we have we obtain decrease also in resistance. This is the final objective, of course. Then we also collect resistant isolates, and we try to understand what happens in the veterinary sector. So 2 examples here, for example, MRSA. We collect MRSA from different, not only from companion animals, also for food-producing animals. This is the example in companion animals. You can see that, well, in horse, it's the specific epidemiology of MRSA, different from cats and dogs. You have very important messages, like, for instance, in cats and dogs, you may know that MRSA, in cats and dogs, mostly reflects the human epidemiology. So it's also a nice message to say that it's not the same direction. It's not always from humans, for animals to humans, but sometimes from humans to animals. And I must say in cats and dogs, mostly from humans. This is also a kind of political message. We also investigate nosocomial infections, whereas the same clone is circulating in farms. This is in a vet clinic in dogs. This is MRSP, medicinal resistant staph pseudointermedius, with the same clone. So that, yeah, this is how to get knowledge on what is circulating in animals and to compare with the human sector in particular. And we provide technical support to local laboratories. And these are the main ones. And the first one is ring trials. They love ring trials. They need ring trials. They need to say, okay, when we perform an antibiogram, is the correct answer. So we receive strains from ANSES, which are anonymized. We don't know the results before, and they correct them. And we are not, if they make mistake, we go and support them. We provide expertise. We don't blame them. We teach them. And that's very important to have, the agreement is based on that. So the ring trials are very important for them. A hotline as well, so in email, we have this type of antibiogram, blah, 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 blah. Training is important, on-site or in our lab. When the staff is changing, where there are new things happening, where something, we perform antibiograms every day, so they can come every day. They just say, okay, we are more experts on gram-positive, less on gram-negative. Could we spend 2 days in your lab and see how it works and ask questions and so on? Yeah, you can do. And we can also sometimes go in your lab and see and teach people. We provide them an annual conference, a meeting with the different labs, very, very important. And sometimes we have common project with them. So just I will go rapidly on ring trials. This is an example of scores. We also check their quality controls in the lab for outliers from say lab. Say, okay, what happens here? You are not in the right position for your quality controls. So this is an example on staff. This is the result of the correction of the ring trials. Lincomycin was not tested. They were correct, but was wrongly transcripted. And this is what we produce a report for each laboratory individually, a common report with anonymized data. And we then produce an oral presentation of the data each year at the annual conference. This is also an example. Only 2 labs have not identified an MRSA. 1 lab has not tested. This is quite concrete for them. And they can change the antibiograms, the disc, whatever, and adapt and progress. We train them on specific phenotypes, like SPRs or whatever. We provide them with updates on standards, where everything technical, everything they need from a technical point of view for their job, for their daily job. This is an example of this is the last program of the meeting. Well, the meeting is a mixture between science, new data, updates, technical things like technical points, the data on ring trials. We also invite, we have an invite speaker from just to open their minds to something else. This is one from Didier Oquet on the environmental component or the One Health component, but we have a lot of friends in the human medicine and and whatever in AMR. So each time we try to invite a colleague or friend to provide a new view to highlight different things. And this is very important also for the network to have this kind of event. So just briefly, 2-3 slides to say that because we think it's highly important, we had the idea to take the lead to initiate such a kind of system at a European level. Not to duplicate Resapath. It's not the question to duplicate it. It's just the question to promote the surveillance of AMR in diseased animals to some extent. So we do that through a European initiative called ESVET, where ESVET is in comparison with ESNet in human medicine. This is the system we produced, where we wanted to initiate it as a proof of concept. It was through the EU JAMI 1, this European joint action, was really a proof of concept. So the job was done by Jean-Yves Madec, you may know. He was under my supervision at the time for 3 years. And he did traveled a lot, visiting everyone in Europe. He was a very nice postdoc for a young, just visiting people and discussing. It was not laboratory, but it was highly useful. We produced a couple of papers. The first job was to map the existing national surveillance system, whether there was a system, which one, no system, how it was working, and so on, across Europe on AMR in diseased animals. So yes, you have very different situations. That's okay. We also try to propose a common vision for ESNet. We also try to define what should be inside of ESNet, what would be the scope of ESNet. And we tried, this was the job of Justine, to have a first ESVET pilot study on data. And as the contributor of this pilot study were those countries. And now we are in the EU JAMI 2, and this second, much more ambitious, much more financed as well. And well, the global objective is to switch from the proof of concept to the full scale, if possible, full scale or implementation of that kind of system. So I will not go into much detail, but the objective, the global objective, is to try to push forward a coordinated system at European level to collect data on AMR from diseased animals. And of course, you know that the situations in the countries are different. There is the production system are different. The animal species are different. So it's also a challenge. But we think it's highly important to have both healthy animals from the food safety issues, but also AMR in diseased animals. When we're talking about national action plans, we need to be closer to the use of antibiotics and to the infections. Okay, so if you want to know more about ESVET, you have some here links. And I will finish there, just to be not too long, take 1 or 2 questions, if any. I would like to thank you for your attention, and just to tell you 2 things, important things. For ESVET, so Rodolphe is not anymore at ANSES. He was at FAO afterwards in Denmark now. But Lucy is the contact person who has taken the role of coordinating ESVET. So please contact her if you have any question on ESVET. And of course, I'm talking about Resapath today, but I'm not alone. It's a very important team, and thank you. This is also the occasion to thank officially all the different persons at ANSES, but also the different peripheral laboratories contributing to Resapath. This is a group effort, and here you have the different people in our institution involved in Resapath. Okay, thank you very much. Hope it was useful. Yeah.
Are there any questions here from the floor? People that attend via webinar can also ask their questions in the chat, and then we will ask them. Any questions? Yes, there.
Jeroen de Wolf, Ghent University. Jean-Yves, thank you very much. It was a fantastic presentation, very interesting. I had 2 different questions. The first one is with regard to the data collection for the Resapath. Is it, of course, it's a project that's running already for many years, but we're trying to do something mini version in Belgium, but we have lots of difficulties with the type of data that is collected and stored by the different labs, all in different ways, different organization. to harmonize that? Do you have a strategy, or do you have had a strategy to harmonize that? Do you have specific guidelines which the labs have to use to provide the data?
Well, this is a great challenge. The challenge is to have the most complete information on the data, because if you just have antibiograms and the animal species, is not sufficient. So we introduced an indicator of how we consider that the data are complete. I mean, what should be inside to consider that we collect what we want to collect. And it's difficult because sometimes we use proxies, for instance, cattle. The proxies for diarrhea in calves is the proxy for diarrhea is the age of the animal. So sometimes we cannot be so sure when animals are moving, to which there is an issue with the geographical distribution and the infranational distribution of EMR. This is also in One Health perspective, because very important in that perspective, because in human medicine they are able to produce data at local level, and we are not so it's not so easy for us through Resapath, because pigs and poultry are mainly Brittany, but also we know that calves move from one region to another one. So yes, the point in my view is to try to make the labs much more aware of collecting data from the vets. I mean, and also what we would like is to have kind of feedback from the vets about the treatment and to connect all of this. So it's not a perfect system, of course. It's a passive system, and we are depending on what the labs are collecting in types of data as a type of data. But there is the strategy is to push them to have the more detailed information on the samples, the metadata. But I would not say that we have a specific strategy, but are fully aware of that point. Yeah.
Thank you.
I don't know if it answers. We can discuss at coffee break, but yeah. Epidemiology is highly important. It's not on. Yeah.
Hello. What about the methodology? In Belgium, so far, it's mostly the diffusion method that is used in first-line laboratory, while for the monitoring, it is the Brose microdilution. Is it the same in your country, and how do you deal with that? In some cases, for specific strains, you are confirming by microdilution, or what is your strategy about the methodology?
Okay, the historical strategy was to have one method, and the one method was the one that vet laboratories use, and they use disc diffusion, all of them. So it was the case for until 5 years ago or something. One day, there is a big company you may know selling systems for MICs and microdilution. Wanted to invest the vet market and to implement microdilution in those kind of laboratories. For now, they didn't success to do that because the volume or whatever is not in currents of that. But we, as Resapath, we were in capacity to collect also MIC data, if any, and we will try we started to think about how to combine two different origins of data, if any. But in the end, we are still on distribution data, so the problem is not on the table for now, but we are fully for SVET is a real issue because some countries monitor AMR in diseased animals only, most of them, I would say, with the MIC and microdilution. So it is a challenge. Yeah, we need to. But well, I think it's a challenge. Yes, but for instance, in hospitals in humans, they use microdilution for bacteria growing rapidly, like urinary tract infections. And they also use this diffusion for some difficult bugs, like Pseudomonas or whatever. So, well, I think we just have to do that properly. But, well, I don't see any major difficulty to combine data originating from different methods.
Okay, thank you very much. There is also a question from the audience that is following via webinar. Benedict?
Yeah. Yes, I have one question. Question is coming from Abu Bakar Garba. AMR is a global issue. Do you have any plan in the future to extend your work outside Europe, for instance, to Africa?
Yes, well, that's an interesting question. We have an action in Africa in cooperation with African colleagues. We are heading the reference center for FAO and AMR at ANSES, and there also a reference center for FAO on AMR in Dakar, Senegal, at the Pasteur Institute. So we are currently collaborating to put in place kind of ring trial or something, distributing isolates to different countries with the specific objective to collect data from diseased animals. So this is on the way, and I think it's highly important to also to harmonize the methods and the expertise in other countries, and especially in countries where MR is a big issue. Yeah. So we try to contribute to that.
Mm-hmm. Yeah, there's another question.
There's a question from Thomas Janssens. What kind of governance is in place to decide on the contents? For instance, which strains, the antibiotics used, the cutoffs of the antibiogram, and how does surveillance influence future antibiogram decisions?
Well, we start from the beginning. When you test an E. coli and you are in veterinary medicine, you need to test a certain list of antibiotics. What is the stuff? There is another list of antibiotics. It's common sense. It's a consensus between microbiologists, epidemiologists, human medicine also. What we do not do is to put or we do not tell them to test for human antibiotics. And this is also a gap, because this system is focused on veterinary medicine. So for instance, we know that in France there are some resistance to carbapenems circulating. We detect regularly OXA48 as a carbapenemase circulating in companion animals through Resapath. But the labs do not test carbapenems. And I think it's a good option, because we do not want to push vets to use human antibiotics, and in particular those ones which are totally forbidden for veterinary medicine. But from an epidemiologic point of view, we know that some very important resistances are circulating in animals, and this system is probably not the best to detect them. And what we do is a complementary way to collect those type of data. We provide, as under the project mode, we provide a disk of carbapenems to the labs to test, to have a first idea of the landscape of what is circulating as isolates resistant to carbapenems within the frame of a project, but not as a routine activity. So this is how the consensus is we discuss with it's under the governance of Resapath that we decide, and also the ministries, because it would not be a good idea for the Resapath to test for vancomycin, for all those antibiotics that are strictly forbidden in the law. But we know, in a One Health perspective, that we need to have the information on what is circulating in animals, even we're talking about resistance, important resistance in humans. So, yeah.
Okay, there's time for one last question. Yes, Benedict?
Question from Philip Boyen. How do you finance the ring trials and the training of the peripheral labs?
how we finance?
Yes.
Yes.
Okay, the money. It's free. No, it's a win-win. So everyone makes part of the way. We provide. Yeah, it's free. I mean, we pay for the transportation. It's kind of it's a cooperation. So there's not a specific budget. They agree to spend some time to do the ring trials, so they dedicate some resources for doing some antibiograms on a set of isolates sent by ANSES. They spend some time to. This is a combination of in-kind resources. There's no money dedicated to Resapath. This is an effort from the two partners, from the labs and from ANSES.
Okay, thank you very much for your presentation and the discussion.
Thank you very much.
We move on to the next presentation, short presentation. They were selected from the abstracts that were submitted, and the next speaker will be Hanne Debergh from Sciensano, and the title of her presentation is Pulmonary Aspergillosis in Humboldt Penguins, Susceptibility Patterns and Molecular Epidemiology of Clinical and Environmental Aspergillus fumigatus Isolates from a Belgian zoo. Data from 2017 until 2022. Hanne, the floor is yours.
Thank you. Okay, good morning, everyone. So last year I was here also talking about the PhD I'm currently doing, which is the Missing Link project. But last year I was talking about Klebsiella. So Klebsiella pneumoniae is one part of the PhD I'm doing. The other part is Aspergillus fumigatus. For both pathogens, we adopt the One Health approach. So using the animal, human, and environmental side and looking at the transmission of AMR between those. So for this one, in Aspergillus fumigatus, we only focus on diseased animals. Then we also included clinical aspergillosis cases from the NRC in Leuven. And then the environmental part is quite big in Aspergillus fumigatus because that part is considering the resistance development is quite big or quite important for Aspergillus fumigatus. So here we included wastewater, agricultural samples, so from wheat samples, horticulture. We also went to several compost companies to select or to collect data. And then also we went to the zoo environment in Belgium. And this presentation will be about a combination of diseased animals and then the environment where they live. So why avian aspergillosis? So aspergillosis is actually the main fungal infectious disease threatening wild and captive birds. And 95% of those aspergillosis cases are caused by Aspergillus fumigatus. And the reason that we did this study was actually that we were contacted by the zoo to perform some environmental sampling because they saw really high death rates of those Humboldt penguins. They already performed several measures. I will discuss them later. But then we went in and performed some environmental sampling because they suspected it had an environmental cause. We performed, of course, some laboratory testing using clinical, data from the penguins, and then environmental samples. And then we looked at transmission of resistant isolates between the environment and the animals. So when we talk about a high rate of avian aspergillosis, what is that really? So in 2017, does that work? Yes. In 2017, they started with about 100 animals, so 100 Humboldt penguins. And if you look at 2022, they have still 28 animals. So about 70% of those animals died in those 6 years, which is, of course, quite high. And for example, in 2018, if you may recall, that was a really hot summer. About 50% of the population died that year, mainly in the summer due to the heat. And that is, of course, because, well, Humboldt penguins, they do not live normally in Belgium, so it's not their normal environment. But then if we, for example, look at the number of aspergillosis, so confirmed aspergillosis, then we see that in 2017 it goes up to 86% of those death cases that are due to aspergillosis, which is really high, of course. What you also see is, so for example, in 2018 you had 37 deaths, but then in 2019, 2020, it really lowered. That was because they stopped with the breeding program. And then in 21, they introduced it again in 4 couples. And again, the death rate rose. 27 of those cases are included in this study. And then if we look at why Humboldt penguins, there is, as I said, the environmental part is quite important. So we have actually 2 main issues here. So one is the environment. Aspergillus fumigatus, it's present everywhere. Each one of us inhales about 100 spores every day. For most of us, it won't be a problem, only if you have a compromised immune system. The zoo is also located near agricultural fields, where a lot of azoles are used to use for crop protection. And then also, of course, the weather. We have quite a humid, especially this year, weather, but then in the summer it can be quite hot. So this also has an influence on the concentration of Aspergillus fumigatus in the environment. And then the other part is the anatomy of those birds. So if you look at the figure, the way between that they inhale air and then to the air sacs, it's quite short, so to say. There is also no mucociliary clearing, so it impacts quite easily in their air sacs, and that's also where we see most of the infections occur. And then the last part is the captivity. It's not their normal environment. It causes a lot of stress on the animals, so of course that has an influence on their immune system. Aspergillosis can be, there are 2 types of aspergillosis. So the one is the acute aspergillosis, the other one is a chronic. So acute aspergillosis is usually seen in young animals and is caused by a very high, sudden, very high concentration of spores. And death usually occurs within about 7 days, so quite fast. And it presents itself with anorexia, dyspnea, so they have problem with breathing. They start breathing with an open beak, and it also has effect on their, they can get depression, so to say. Whereas if you look at chronic aspergillosis, this is usually due to a prolonged immune depression, which is related to their captivity, because it can give stress, but it can also be related to their husbandry. Here we see a death between, after 1 to 6 weeks after exposure. And then here we see some weight loss, also changes in behavior and activity, but also they start coughing, and they really have trouble with breathing, which is quite sad to see. As I said, most of the chronic cases happen in older animals and is related to their immune status. And if we look at the animals that are included here, the youngest one was 9 months, but the oldest one is 22 years. And most of them are over 10 years of age. So the measures that they already took to help with reducing this problem was, in the summer they monitored the water temperature, so it stays below 20 degrees. And this is related to where they usually live. So they live close to the Humboldt current, and that is quite cold water. So that's why they try to keep it as low as possible. They also improved husbandry, so they used plastic nests instead of wood, because that could be a place where Aspergillus was growing. They provide some shade in summer. And as already mentioned, they stopped or lowered the number of breeding programs, because that really has an effect on their immune system. And they also stopped the direct contact with visitors. Furthermore, if they see that a penguin is suffering, they will try to, they will give prophylaxis, so they use itraconazole, an azole, and it usually is given for about 3 weeks. But some penguins in the zoo, they receive Sporanox daily for a long period of time, and that's mostly the older animals. As I said, we performed some environmental sampling. So we performed 4 types of samples. So we included air sampling in the nests depicted by the red cross, then water sampling, sand sampling, and that was in the nests that you see located there, but then also where they get their feed. And then also we took some swabs of some nests that were not there were no animals breeding there because we could not test or sample those nests where penguins were breeding due to stress that it would cause. We performed this sampling 4 times, so 1 time per season. And then, this is a picture. So you see there were the nests located. Then you see the penguins, which are nicely separated from the visitors. In the lab, so we performed air sampling, sand sampling, then the water was also filtered, and we took some swabs. All of those samples were placed on 2 media, so in multichloramphenicol, so just a normal or a standardized medium for fungi, and then we also seeded them onto multichloramphenicol and tibuconazole. So tibuconazole is an agricultural azole, and we use it as a screening tool for resistance against the medical azoles, because there is some cross-resistance between agricultural and medical azoles. That was incubated for 48 hours at 48 degrees to limit the growth of other fungi, for example, Mucorales. Then only those colonies that grew on the MC plus tibuconazole were identified using the MALDI-TOF MS. Then antifungal susceptibility testing was performed on all those isolates, and if we saw isolates that were resistant to at least one of the medical azoles, we also performed sequencing of the CYP51A gene to look at their mutation. So the contamination routes, in nests, we only observed 1 colony. We did not observe any in water or sand, but we observed a high concentration of spores in the air. So what you see on the left part, so the MC, this is reflecting the, you would say the normal, so not, there was no tibuconazole present, so there was no selection there. You would expect to see higher concentrations in the fall compared to the summer. This is not the case here, and this was due to the contamination on the plates. So there was a very high load of other fungi on it, and we were not able to isolate those colonies. So on the MC plus tibuconazole, we isolated 64 Aspergillus fumigatus colonies, and those were all subjected to antifungal susceptibility testing. On the top part of the table that you see here are all the environmental samples that showed phenotypical resistance. So that was 8 out of 64, so we observed a resistance rate of about 12.5%, which is quite high. And all of those were pan-azole resistant, so resistant to all of the medical azoles, so voriconazole, itraconazole, isavuconazole, and posaconazole. And then you see on the right part of the table the mutation. So those were all the TR34L98H mutation, which is known to originate from the environment. So in Aspergillus fumigatus, resistance can occur either through the patient route, so they say, so which is the same as in bacteria. You give a patient or a penguin, you give it some azoles, and then due to that stress, it develops resistance. But in Aspergillus fumigatus, there is an important part of the environment, because we see cross-resistance occurring due to the use of agricultural azoles for crop protection. And this mutation, so the TR34, but then also another mutation with tandem repeats, so the TR46, is actually linked to development of resistance in the environment. And then if we look at the veterinary part, here we have 6 out of those 27 that displayed azole resistance, which is really high, so 22%, because I'm finishing a manuscript also on veterinary aspergillosis in other birds and mammals, and there we have a resistance rate of about 3%. So it's a really big difference. But the important part here is that we also see the TR34 arising here, so the environmental resistance. This penguin, it did receive Sporanox since 2017, so over a year it received treatment, but we still see the TR34, so this is also in line with other data or other articles that treatment is not the, so azole treatment is not the main resistance part in veterinary cases. As a last part, we did genotyping of a subset of species. So 45 Aspergillus fumigatus isolates were genotyped using the variable number tandem repeats, so VNTR. And then on the left part, so A, you see all the isolates depicted by a circle, and the line between those shows the proximity of those isolates between each other. In gray, you see the environmental parts or environmental isolates, and in orange, you see the veterinary isolates. In general, they're not closely related at all. They're quite far from each other, but we do see one cluster with 6 environmental strains and then 1 veterinary strain. And then if we look at the right part, so C, then you see that that cluster is a cluster also of resistant isolates. And all of those carried the TR34L98H mutation, which links to an environmental contamination of the penguin. So as a conclusion, we saw a death rate due to aspergillosis not up to 70, but up to 86%, but almost 70% of the penguins died in those 6 years. We did detect a high environmental load in air samples. And also the presence of the azole-resistant Aspergillus fumigatus in the environmental and in clinical samples was detected, which really highlights the importance of the One Health approach in this case due to the importance of the environment here. It's also the first detection of a TR34L98H mutation in a Humboldt penguin. And then genotyping, this was not shown here, but there was one isolate, so from a penguin, where we had 2 isolates from the same penguin, isolated at different times, but it did have a different genotype. So this also shows that, as we see in clinical cases in humans, that a penguin can be infected with 2 types or more strains of Aspergillus fumigatus. And we also observed a clustering between environmental and veterinary isolates. So thank you very much for your attention.
Thank you very much, Hanne, for this interesting presentation. Are there any questions? Yeah, there's a question from the audience at home. Benedict?
Yes, there's a question from Alessio Gianelli. Were penguins tested for toxoplasma?
No, we only focused on Aspergillus fumigatus.
Okay, Jeroen?
Have the results of your study influenced the treatment strategy of the organization of the zoo in any way?
So we didn't follow up, but the treatment regimen, I don't think they will change it, because here we did see that the main source of contamination is the environment. And the treatment doesn't really have an effect on the selection of resistance or the occurrence of resistance. So it's really their environment that they should change. Because, for example, in the same zoo, they also have a different type of penguins, and they are enclosed, so really they are not exposed to the environment. They're also HEPA filters and so on, and there they do not see that problem. So it's really their environment and not really the treatment that they receive.
Okay, still time for one last question. If there are no further questions, thanks again, Hanne, for the nice presentation. And we move on with the last presentation before the coffee break, and that presentation will be given by Jacob Diderich from the University of Liège. And the title of the presentation is Stability and Lytic Activity Assessment in Milk of Bacteriophages Targeting Escherichia coli Causing Bovine Mastitis. Jacob, the floor is yours.
Thank you. So, good morning, everyone. It's my pleasure to show you my work on the stability and the lytic activity assessment in milk of phages targeting E. coli in the context of bovine mastitis. So, as you probably know, bovine mastitis is an inflammation of the udder that is caused by many bacterial species. And in my work, I focus on E. coli, which causes environmental mastitis. So these strains are in the environment, and then they can infect the teeth and disseminate in the udder, and that would lead to a strong inflammation with all the clinical symptoms associated to this. And we also have as a cause a diminution in the milk production, and that is responsible for almost the half of the cost of mastitis caused by E. coli. So the treatment of mastitis is actually more symptomatic treatment, so administration of NSAID, frequent milking, and feed therapy. But if there are general signs from the cow, then we would apply intramammary treatments. And so these treatments, as you know, they would cause resistances, and that's why we would like to find some alternatives to it. And so, as you probably know, there are huge efforts that are made in Belgium. So here are numbers from the AMCRA. So we know that there are huge efforts and a big decrease of the use of antibiotics, but on the other hand, there will always be mastitis, so it's important to find some other treatments. So in my work and in our lab, we focus on the use of phages as complementary treatments to antibiotics. So phages are viruses, and they are specific to prokaryotic cells. So that's a good point for us because they only target the bacteria. They are very prevalent on Earth. So it is said that phages are the most prevalent microorganism on Earth, and so there is a lot of diversity of them. Phage, they are viruses, so they can co-evolve with their bacterial host, and so sometimes the bacteria become resistant, sometimes they are prophage that are introduced in the bacteria. Sometimes we can also have a coexistence between the phage and the bacteria, so we have like a dynamic population. And so as a conclusion, we can say that the relationship is a bit complicated. But if we focus on other advantages of phage therapy, I said that they are specific to their bacterial host, so they will multiply and do their cycle at the site of infection, so in this case, in the udder. As we want to make an intramammary treatment, we have to be sure that the phage would be stable and efficacious in the udder environment. So that means in milk and also all the components that are present in milk. So there were three main objectives of my study. First, we isolated phages targeting E. coli causing mastitis. And then we characterized the phages and we made in vitro assessment of the phage efficacy. First, we had to collect bacterial strains, all isolated from mastitis, and we collected 50 strains from Arcia, and 3 new strains were isolated on the field in our lab. 6 strains of our collection were ESBL. After that, we collected some wastewater samples. So here we took 9 wastewater samples and 1 slurry sample from a farm because these environmental samples, they are sources of potential phages. And we put the phage in contact with different bacterial strains from our collection. We put them together with a bit of growth media and we let it incubate. And if in our environmental sample there would be one phage or phages that are specific to the strains that we added, then the phage will multiply itself and eventually it will lyse all the bacteria of our sample. So we will see a clear lysis in our tube. After that, if we see this lysis, we centrifuge the tube, we filter it, and we make purification to be sure to have pure mutants of our pure clones, of our phages. So this method allowed us to isolate 10 new phages that we characterized for our application. So this characterization, basically, it's a bit like an ID card. So we want to know how the phage behaves, what bacterial species it can infect, its stability in different conditions, and we also want to know if the cycle of the phage is lytic or lysogenic, because for phage therapy we don't want at all to have lysogenic phages. So first we made our host range. So we took all the strains from our collection, the bacterial strains, and we put them on an agar to make an overlay. And then we spot all our 10 phages on this bacterial overlay. And after incubation, we look if there is lysis or not, and we characterize it. So sometimes if we are unlucky, we see that there is no lysis. Sometimes we have some small lysis plaques. We can have opaque lysis, semi-confluent lysis, and confluent lysis. And here they are the results of our host range. So we have our 10 phages. And here we have the percentage of lysed E. coli strains of our entire collection. So we see that we have a lot of variation. but we see that this host range is quite narrow. So there's just this phage that targets almost 30% of our collection, but all the other phage just lies a small number because they would be specific to some strains. And so we checked in the literature, and for the first step of the phage cycle is the attachment of the phage to the bacteria. And for E. coli and gram-negative bacteria, the first obstacle of the phage would be the lipopolysaccharide and the O antigen of this LPS. And so, as you know, these O antigen are very variable, and there is a lot of diversity of them. And so the phage has to be very specific to this O antigen of the strain to be able to start the infection process. So that would explain the narrow host range of our phages. After that, we checked some different conditions to see if our phage would remain stable. So there were pH and temperature stabilities. Here we took our 10 phages again, and we put them for 1 hour in PBS adjusted at different pH, ranging from 2 to 12. And then after 1 hour, we make a titration to see if there is a change in the stability. And so here we see that from 4 to 10, there is a really good stability. So we don't see any change in the titer. But at more extreme pH, there is a total decrease because the phage cannot remain stable. And we have just one phage here that remains stable at a pH of 12. Regarding temperature, it's the same process. So we put the phage for 1 hour at different temperature, from 25 to 60 degrees. And we also make titration. And we saw that from 25 to 45 degrees, we have a really good stability. And at 60 degrees, we have a strong decrease. And some phage remain stable, but with a few logs of a decrease. But what interests us is its ability to remain stable in the mammary gland. And so with these parameters, we know that all our phage would be suitable for an intramammary use. After that, we wanted to check more in details if these phage were suitable. So we made a few assessments of the phage in milk. And so therefore, we took raw milk. So we milked a cow with a low risk of mastitis. And we checked with the CMT to check the absence of mastitis. And then after that, half of the sample was heat treated at 70 degrees for 35 minutes. And then we make a stability control of the raw milk and the heat treated milk. And why did we take heat treated milk for our experiment? Is because there are a lot of proteins, immunoglobulins, and so on that could interact with the phage. And so we wanted to know if there is an impact of these heat sensitive proteins on our phages, because we know that for staphylococcal phages we have these effects, and so we wanted to know if it's the case also for these phages. So first we make a stability assessment. So we put the phage in the raw or the pasteurized milk. We let it incubate for 6 hours, and then we make also a titration to see if there is an evolution of the titer. We saw that for all our phages, they were all stable, and when we make some statistical analysis, we looked at, and we saw that the pasteurization, the heat treatment, didn't have any effect on the stability of the milk. So these proteins cannot affect the stability of the phages. After that, we made an in vitro efficacy assessment of the phages in milk. So we also had samples. We inoculated the phage and the bacteria. So the phage could replicate because we have the bacteria, and the bacteria would be lysed by the phage. And we also had a control with only the bacteria, without phages. We chose a MOI of 0.01 because it was the optimal MOI for the phage multiplication. And then we inoculated it, and we made some bacterial titration after 1 hour, 3 and 5 hours of incubation to see if there is an evolution in the bacterial titer. So for this experiment, we took again raw milk, heat-treated milk, and also UHT milk, which is milk from the supermarket. And we did also for our controls. So here we have our 10 phages, and we see that we have some different pictures, different results. If we look at the first phage, the L819, we see that we have the line with the dots. It's the titer of the inoculum. And we see that in the control group, so just the bacteria, there is a multiplication of the bacteria. So that's normal. And with the phage, after 1 hour of incubation, there's not much going on. But after 3 and 5 hours, we see a complete decrease of the bacterial titer with the presence of phage. So that's a very good case. That's really what we want. But unfortunately, for other phages, like here, the N824, we see a strong decrease with the phage. But after 5 hours of incubation, we see that there is a bacterial regrowth. So that would mean that we have some resistance, and that the phage is not sensitive to the phage anymore. The bacteria is not sensitive to the phage anymore. But still, the good point was that after these 5 hours, we still have a big difference between the control group and the treatment group. For other phages, we had some intermediary results. So sometimes we see a small regrowth. Sometimes, yes, it remains stable. So all phages had different behaviors. So as a conclusion, we can now fill our ID card, not completely, but most of it. So I took one phage, the L819, which I showed you. So it was isolated in slurry. It had a narrow host range, like all the other phages. It was stable at a pH from 4 to 10 and at 25 to 45 degrees. They were stable in milk, and it has lytic activity also in milk. But yet we don't know if the cycle of the phage is lytic or lysogenic, because we have to make genome sequencing for it, and we didn't have the result yet. So that's our perspectives, all this sequencing part. So we will analyze the genomes of our 10 phage. So we will know the taxonomy of the phage. We can make phylogenetic tree and alignment with other phages to see if there are some mutations, if there are conserved regions, and yes, between phages from the same genus. And then we annotate the genes, and we can identify if they are lytic or lysogenic genes. And so after that, we can say if the phage is lytic and suitable for an intramammary use for a treatment. We also want to make sequencing of our bacterial collection, because as I told you before, this host range would be linked to the serotype and maybe other receptors that are on the bacterial cell. And so the sequencing will give us this information, and after that we will try to associate the host range with all our data that we have, and explain why some phages can lyse certain bacteria and some other not. We would also like to make a characterization more in depth, and that's more for our resistances that we identified. So we want to isolate these colonies and look also if they have different genomic profiles than the wild type strains. So thank you for your attention. Thank you for my lab and my promoter, Damien Thierry, and all the other people that contributed to the work. And thank you for your attention. Don't hesitate if you have any question. Thank you.
Thank you, Jacob, for this interesting presentation. Questions from the audience on bacteriophages?
Yes. Hi. So, thank you very much for this job. How do you see it concretely on the field? Do you think you would need to test something, I mean serotype or the efficacy of the phage before using the phages, or will you go blind and see if it works? Or how do you, the host range issue? How you think you would deal with that?
Yes. So in the case of bovine medicine, so it's medicine on the field, the best would be to make milk analysis to isolate the strains and to make a host range, but that would take a few days. And so maybe we could make a blind treatment with a cocktail of phages, so different phages that have different host ranges, and to put that in first intention, and then in parallel, like for antibiotics, make an analysis in the lab to know if the selection was good or not.
Yeah, there's a question over there.
Okay, Etienne Thierry, University of Liège. So thank you, Jacob. That's of course very important to test phage therapy and potential phage therapy as an alternative to antimicrobial treatment. Of course, to test phage activity in face of milk is of course very important for this kind of udder treatment. My question would be the quality of the milk in inflamed udder. Okay, we can assume that in a normal milk, I would say the antiviral peptides, lactoferrins and so on, would be roughly at the same concentration, but due to the inflammation, do you think that the milk inside the udder would have the same, let's say, concentration of these antiviral peptides? That would probably make something different based on your results, which needed to be done, of course, using normal milk.
Yes, so indeed, it's a good question. And so, but for our characterization, we needed to have healthy milk for the standardization of our results. But it is true that milk from mastitis has a different composition, as you say, the lactoferrin, lactoperoxidase, and so. It is described that they can have an effect on the phages, and so I think that they could interact with it, and so it would be interesting to make the same experiment with milk from mastitis. Yes, it would be interesting.
Okay, other questions? Yeah, Benedict.
I have two questions from people following at home. First one is from Marilyn de Souza. How do you see the application of these phages in field conditions?
So once all this characterization and all this authorization are okay, I think as I show you, there are a lot of resistances that can appear really fast. And so what is usually performed also in human medicine on the field, what is also already done, is that then they combine the antibiotics with the phage, and so we really have this complement of both, and so that would be like normal administration of the preparation, but in the same time the phage and the antibiotic.
And the second question was metagenomics and next generation sequencing important in this study?
Yes, so I don't have the results yet, but the sequencing is really important. As I said, for the determination of the cycle of the phage, the multiplication cycle, because we want to be sure that we have lytic phages, because with temperate phages they can transfer resistance genes and virulence genes to the bacterial strain, and so we want to avoid it, of course. And all this sequencing is also important to to be able to classify the phages and to be sure for cocktails, for example, to choose a broad panel of phages.
Okay. Other questions?
There is one other question from home, from Julie Bellemans. Why choose for bacteriophages instead of endolysins?
We could also use endolysins, and it's also been done in our lab. It's currently in progress with my colleague, but it's different techniques, and yes, of course we could also choose these endolysins, but it's different subjects, and both are very interesting. Yes.
Okay, thank you very much, Jacob, for the discussion.
We will start the second session of this symposium, so it's my pleasure to chair this session, and we will start this second session with a presentation from Zoë De Mol. So Dr. De Mol is a veterinarian working as a PhD student in the Department of Internal Medicine at Ghent University, and her talk will give insights about the determinant of antibiotic prescription behavior among Belgian veterinarians. So, Zoë, the floor is yours.
Okay, I think my slideshow is going automatically. That's not... Yeah. It's okay. Yeah, that's okay. Okay, good morning, everybody. Big thank you for giving me the opportunity to present my work today on the prescription behavior of veterinarians. So, how do veterinarians decide whether to prescribe antibiotics or which antibiotic to prescribe? The answer to this question is crucial in combating antimicrobial resistance, or AMR. But why is this crucial? AMR needs a One Health approach to safeguard effectivity and availability of antibiotics in both human and veterinary medicine. As you may all know, the use of antibiotics is one of the major drivers of AMR, and antibiotics are being used in both humans and animals. That's why optimizing the use of antibiotics is necessary in both sectors. In the past decade, several initiatives have been installed to promote responsible AMU in food-producing animals. For example, the legislation on prophylactic use of antibiotics, the legislation on critically important antibiotics, but also benchmarking and data collection. From the 1st of September 2024, the use of critically important antibiotics in companion animals will also be restricted. That's why the aim of our study was to explore the determinants of antibiotic prescription behavior among Belgian companion animal and livestock veterinarians. We wanted to achieve this goal by using qualitative research methods, and we organized focus groups. During a focus group, we bring together a small group of people to answer questions in a moderated setting. This allows for an exploration of different experiences and perceptions of prescribing antibiotics. The focus groups were organized online and audio recorded, transcribed verbatim, and then analyzed inductively, so an inductive thematic analysis. This means that we really start from our data, we don't use a predefined framework, and then we assign codes, subteams, and teams to the passages in the transcript. So we recruited 42 participants, of which 19 companion animal veterinarians, and I'm sorry, there is something wrong with my animations. So I recruited 19 companion animal veterinarians and 23 livestock veterinarians, of which 5 pig veterinarians, 11 ruminant veterinarians, and 7 poultry veterinarians. We aimed for variety in We aimed for a variety in participants and a diversity in age, experience, geographical location, but also position within the practice. As you can see in this table, we organized 5 Dutch-speaking focus groups, 2 French-speaking focus groups, and 1 French-speaking interview. So if we have a look at the results, data analysis revealed 6 overarching themes describing the prescribing behavior of Belgian veterinarians. So Theme 1, the diagnosis. Theme 2, the therapeutic options. The third theme, veterinarian characteristics. The fourth theme, client characteristics. The government initiatives to promote responsible AMU, and veterinarian client communication. Next, I will provide a small summary of each theme, illustrated by quotes from participants. So as you can see on this slide on the right, you can see 2 quotes from participants, and they indicate that the clinical examination is actually the first step in the decision-making process to determine if there is a bacterial etiology or antibiotics are necessary or not. More severe clinical signs, such as fever, will lower the threshold to prescribe. To confirm a bacterial etiology, participants will use diagnostic tools, such as antibiotic susceptibility testing. But as you can see in the second quote, veterinarians will often already start the treatment with antibiotics before the results of the test are available in case of emergencies or time constraints. As you can see in the third quote, the price of these tests is often a significant barrier, especially with economic vulnerable pet owners, but also in livestock. However, the participants said that this allows a more targeted treatment and ultimately saves money. That's why antibiograms were perceived as a valuable diagnostic tool. Moreover, cytology in companion animals was perceived to be very valuable as it requires minimal financial and time investment. If we have a look at the second theme, the available therapeutic options, you can see that it's very important to evaluate the characteristics of available antibiotics. For example, in companion animals, the veterinarians will consider the user-friendliness of the antibiotic. For example, in case of aggressive cats or pet owners being unable to administer oral medication, and then they often will opt for a long-acting single injection of sevoflurane, which is a critically important antibiotic. In livestock animals, the withdrawal period was also noted to be very much significant, for example, at the end of the production cycle in poultry. And this is, I think, very much important that the veterinarian will also decide whether they will use a curative, metaphylactic, or prophylactic treatment. And as you can see in these 2 quotes, some participants say, okay, the preventive use of antibiotics, we no longer do this, while others provide a more nuanced approach and say, okay, sometimes we do it to prevent financial losses, for example. But it was a well-considered decision. Next, participants also noted that they will consider alternatives for antibiotics, as this will reduce the need for antibiotics. For example, in companion animals, they will administer a local antiseptic therapy to prevent the use of systemic antibiotics in case of pyoderma, or they will administer NSAIDs in case of mild clinical signs. And of course, the majority of participants really emphasized the importance of preventive veterinary medicine, and they perceived vaccines to be the most important measure. If you have a look at the veterinarian is responsible for prescribing antibiotics, and that's why their characteristics will influence the decision-making process, which is based on their expertise in prescribing antibiotics. So this expertise is a result of knowledge and experience with prescribing antibiotics, evolves over time, is also a result of trial and error, but is also influenced by education and fellow veterinarians. So as a result, this threshold for prescribing antibiotics varies very much among individual veterinarians. And if you have a look at the awareness of, it goes automatically, so if you have a look at the awareness of veterinarians, the veterinarians that participated, the majority showed great awareness of EMR, and they also indicated moving away from some systematic EMU. However, they also indicated that they experienced that not every colleague has already adopted this new mentality. As antibiotics and the sales of antibiotics and the prescription of antibiotics is a source of income for the veterinarian, reducing EMU is a possible loss of income. But participants also said that this potential loss of income can be compensated by focusing on preventive veterinary medicine, diagnostic tools, and a more individual approach. And the last quote, the lack of time was considered as a very important barrier for this approach, responsible EMU, especially in livestock veterinarians. The client, so the farmer or the pet owner, is responsible for the animal, but also will sometimes administer the antibiotic. That's why, for example, their awareness will influence how much they will pressure the veterinarian to prescribe antibiotics. Participants experienced a great variance in the awareness among their clients. And so they advocated for targeted education and awareness campaigns among pet owners and farmers. As we all know, the animals serve as an income source for the farmer, and prescribing antibiotics is often the cheapest solution in terms of labor, financial, and time investments. However, participants also said, okay, but if you focus on a more preventive approach, this will ultimately save money on the long term. And then if we have a look at the last quote, it's very much important to take into account the financial capabilities of the clients, especially, for example, pet owners, when you decide to use diagnostic tools or prescribe a certain therapy. If we have a look at the government initiatives, for example, the legislation on the use of critically important antibiotics in food-producing animals that has been installed since 2016, participants noted that the majority of participants noted that they have adjusted their prescription behavior in line with this legislation, while in companion animals this will change from the 1st of September 2024, and they said, okay, this changing of my prescription of critically important antibiotics, for example, Convenia, is a barrier. That's why they still need to make this mind shift, while livestock veterinarians have already made this mind shift. And that's also important to say, I think, that the companion animal veterinarians also noted that this new legislation will be a helpful change, as it will facilitate communication with pet owners. And then there were also some participants that noted that the legislation on the prophylactic use of antibiotics and the on-farm stock was somehow unclear for them, and they advocated for more communication or clear guidelines. If we have a look at data collection and benchmarking, participants really experienced that these periodic benchmark reports promote behavioral change among farmers that were previously unaware or uncompelled to change their behavior, to change their AMU. This because they avoid the consequences, for example, exclusion from the quality label. So this is a helpful initiative for them. However, they emphasized that taking into account welfare, the animal welfare is very much important amidst constantly decreasing EMU limits, and they also demanded a fair compensation for the efforts that they have done for data collection and promoting responsible AMU. If we have a look at guidelines, the guidelines are not the first consideration in the decision-making process, as participants value therapeutic freedom. However, they said that it serves as a backup, a backbone for especially young graduate veterinarians. They also advocated, especially in companion animals, more practical guidelines in line with the new legislation on critically important antibiotics in companion animals. If we have a look at campaigns, especially in companion animals, veterinarians noted that campaigns aimed at the general public are very useful to facilitate communication with their pet owners. While in livestock, they emphasized that it's very much important to avoid a negative perception of agriculture when you do a campaign aimed at the general public on responsible AMU. And last but not least, if you have a look at communication, how do veterinarians tackle this pressure to prescribe? For example, they will take their time to provide a very clear and thorough explanation, explain the side effects, why antibiotics are not necessary, the decision-making process, and so on. So this takes time. Moreover, participants will also use their authority as a veterinarian to say, okay, in this case, we'll not prescribe antibiotics. So there's no discussion between client and veterinarian, while others say, okay, but I will take into account the considerations or the ideas of the clients to ensure therapy compliance, which is also important. And you can also see the word trust. I think mutual trust between client and veterinarian is very much important to create an established relationship between, for example, the farmer and the herd veterinarian. Okay. I would like to end my presentation with our main conclusions. So we see that veterinary medicine has really evolved from systematic AMU towards prevention, which is actually facilitated by the past initiatives promoting responsible AMU and resulted in a mindset shift among veterinarians. The decision-making process is primarily based on their expertise. Financial and time constraints are important barriers for responsible AMU. Communication is key to tackle pressure to prescribe, and targeted education and awareness campaigns are very much needed to promote responsible AMU. And what I mean with targeted, I mean, everyone. So the veterinarians, farmers, pet owners, but also the general public. So these insights can be used to optimize and design future, but also current initiatives promoting responsible AMU. With this, I would like to end my presentation. I would like to excuse myself for the mild inconvenience with animation, but yeah, if you have questions, shoot.
Thank you, Zoe, for the presentation, the nice presentation, and the techniques was not with you, but it was nice
okay.
and really. No, the techniques. I mean the animation was not really with you today, but the presentation was nice, so we well understood everything. So, are there any questions in the audience? Yes.
Yes.
Good morning, Zoe. Thank you for your presentation. I'm Bob Ruswans. I speak to some degree on behalf of the small animal practitioner. It amazes me that one of the limiting factors for using culture was the price. Do you mean the price of the lab test or the price that goes along with taking the sample? Because in our practice, it's not that easy to take samples from the lungs or from any organ in the... And I'm not talking about the aggressive cat, I'm just talking about the dog that's in front of me. Is that the price that goes along with it, or the lab price?
In this case, I mean the financial price, so the price from the laboratory, but I emphasized especially economic vulnerable pet owners. So I know that there are pet owners who are very much willing to pay this price, but in general, it can be a barrier. But of course, I don't think it necessarily came out of the data, so from what the people said during the focus group, but I think indeed that the price from taking these samples is a burden, an extra burden for the veterinarian. So in this idea, it can be a cost.
This was what amazes me, that it didn't came out as a hurdle, despite the price, just taking the sample and the word of it. It's really strange. It didn't came out of these working group vets.
But in fact, if you have a look at it, like there is a quote where they say, okay, in certain cases, I will already start my treatment because I don't have the time or it's not practical to take these samples in case of emergencies or if you're on call duty or you don't have the time, and it's not always feasible, especially if it's a life-threatening condition, to take these samples. So if you mean this, yes, it came out of the data.
Okay, thank you.
Yeah.
Hello, Ria Kreve. I'm an animal expert for the Belgian presidency. Thank you, Zoe, and congratulations on the work. I think really nice results. Also very interesting how you worked with focus groups. So my question is more like relating to the methods. Can you tell a bit of how participants were included in the focus group? So also what kind of biases do you already foresee from that process getting into your results? And also I'm interested to see what kind of next steps you're planning to do with this work as well.
So I recruited the participants by actually contacting the available contact information online of veterinarians, so by mail. Also my personal network of colleagues. But there were some newsletters from scientific organizations, professional organizations, such as SAVAP or VEDA, and also in Wallonia I tried the same thing. So I tried everything possible to recruit the participants. Of course, people that participate are extra motivated on this topic, so you can not expect that people that don't care about antibiotics or the responsible use of antibiotics participate in this study. But yeah, that's always a problem, I think. And the next steps is that we are designing a pilot intervention, antimicrobial stewardship intervention in small animal practices, and I will also include my findings in this intervention. And yeah.
Bill van Dal, representing UPV. Zoe, congratulations for your work. I have a practical question. I did not saw in your presentation, at least, were the vets able to speak about their importance of the freedom of choice of antibiotics? Was that part of the questions?
You mean like the therapeutic freedom?
Yeah.
Yeah, that's what I mean with expertise. I forgot to mention it, but that veterinarians prefer the therapeutic freedom above guidelines, and that's why they rely more on their expertise.
Thanks.
That came, the majority of participants said this, indeed.
Okay, thank you, Zoe. I don't know if you have a question online.
Yes, I have one question. Question coming from Alessio Gianelli. Thank you for the excellent presentation. Have you noticed for the 6 different teams variations in relation to the age of the vets? And if so, for which topics in particular?
No, actually not, but what the participants said that they experience that older veterinarians are, for example, less aware. But in my opinion, the older participants were having the same ideas on the need for responsible AMU. However, for example, recent graduate veterinarians often have more insecurity with prescribing antibiotics, and they really advocated for more practical guidance or guidelines. And I think the people who are more experienced will rely more on their expertise, while people that have less experience will more rely on guidelines as a backbone for their decision making. So it can be really useful, I think, for prescribing insecurity, for example, in stressful situations or in case of emergencies. In that case, I noticed a difference in age. And also maybe that younger veterinarians often have more trouble with countering pressure to prescribe from clients because they have more insecurity with this communication with their pet owners. So while older veterinarians have this authority to counter this pressure.
Okay, thank you, Zoe, for the presentation and the discussion. So.
Okay.
So we can move to the next speaker. who is Léonard Théron. Dr. Théron is Chief Technology Officer at Rumexpert. Rumexpert is a company that provides consultancy in ruminant medicine and production management. And Léonard will talk about new monitoring tools that can optimize the udder health management and antibiotic reduction in dairy farming. So Léonard, please.
Thanks a lot, Damien. Hello, everybody. Thanks for this presentation. So Rumexpert is a small CRO, veterinary CRO. In Belgium, we are a startup spin-off company from University of Liège. And today we are just showing some of the tools that we are trying to put in production for the future. The main stakes that we are facing nowadays is that the people that are trying to do the change are actually under heavy load of work, and the data that they need, as Zoe has shown, is not arriving exactly at the good time, at a good moment when the decision making is done. And the decision making needs 2 to 3 different people, different specialty to get together to do the change. The second thing that is interesting is that we are moving in a digital era where diagnostics can be transferred through a lot of different digital way, so that they could be stored, as in the presentation of Jean-Yves Madec, that we can exchange these results quite fastly between different sites. The third stake is that at the same moment we are gathering a lot of data from also the production and subclinical data that are interesting for the future, because biomarkers are in fact analyzed in real time. The cow, as we used to say in Rumexpert, is probably the animal the most connected on the planet, so we can do things about it and use this to do the change. But in the end, what we are really doing in a One Health approach and in the climate change movement is that we are preventing probably some milk waste. So the food waste is the ultimate goal. So antibiotic resistance is one side of the issue, and the food waste is the second one. The food waste solely in Belgium, which is 2% of the European market, is 150 million euro per year of milk that doesn't reach the market because it's lost to antibiotic usage, sick animals, etc., etc. This presentation has been prepared by my data team. So we have a data team of engineers. So we are engineers and vet together, and it worked. Okay, so basically the system that we are using is a integrated platform to gather all the data. It's called SALVE, Système d'alerte vétérinaire pour l'élevage. And it allows user to have multiple farms at the same time. So you can do benchmarking in real time. All the data is gathered through database and sensors. The structure of the protocol is done to be open. So it's open SQL database. So basically we can connect multiple sensors to the database. This is the example of the tank milk analyzers. So for instance, we are just analyzing the tank milk data, tendencies and stuff, and giving away addresses to the team of professionals that are around the farmer and the farmer himself if he needs. We can also analyze the individual data of the cows through the milk control data, for example, and giving away individual or herd approach. And then you can do the same for demographics data, mortality and stuff. So basically for the vets and the counselor of the farmer, it's a complete dashboard that is making you aware of what's happening inside the farm, but also at the environment level, because we can monitor field weather from a network of the CRAW. This approach is done in Belgium, and it leads me to 3 new tools. The first is the analysis of the potential of selective dry cow therapy in a group of cow. The second one is the dynamics of the udder health at the farm level and the link with some existing factors. And the third is what can we say about the impact of nutrition on health of the animals using udder health markers and nutritional markers. The first tool is really simple, okay? It's selective dry cows, so we are algorithmically selecting cows that are available to do no antibiotics. And we base this on existing frameworks that are designed by AMCRA, etc., etc. On our statistics, which are quite limited for now, because in the pilot phase we have 23 farms, but 65% of the cows can be dried without any antibiotics. Basically, it looks like that. So you go in the milk control area, and then you go to the individual list of animals, updated on the last control, for example. You can select the animals on tags that are basically, for example, cows that should be dried because they are too low and they don't need to be producing anymore. You can also select them by risk of disease, and you can also select them by good candidates for selective dry cow therapy. It's like that, it's instantaneous, and you can export the graph to the farmer, etc., etc. So for the vet, without any contact with the farmer, as soon as you have the authorization of the farmer, you can take decision from a distance and help the farmer programming your approach. The second tool is cure and infection rate. Well, what I've been doing during my thesis in the faculty 8 years ago was analyzing the dynamics of udder health and understanding that the cure rates were linked to the nutrition and immunity of the cows, where the infection rate were actually linked to the practice at the farm level. What is interesting is that farms can have different patterns. For example, this is a farm where for 6 months, the cure rates were extremely low, but the infection rate were not high. But the result of having a low cure rate is that even with a low infection rate, you get a positive evolution of incidence of mastitis. But you see that as soon as there's a change in the nutrition, and in this case it was a change in the nutrition, the cure rates got up, and then the herd will solve by herself the issue at the subclinical level. You can have different situations. So this is a farm where infection rates are always high, and cure rates are always low. So it means that chronically, this herd has welfare and probably nutritional health issue that is affecting the global mastering on the udder health. What is interesting is that if you compare this cure rate to the economical losses at the farm, and specifically the part that is avoidable, there is a link between the cure rates and the avoidable loss in udder health. So the more cure rates you have, and we created a composite cure rate that is assessing live lactation cure rates and dry-off cure rates. So it's actively something that can bring money and also bring health to the animals. The third tool is based on the infrared spectrum of the milk. So basically we are analyzing the whole spectrum of the milk and giving away some families of fatty acids in the milk. So we have 3 big families, the preformed, so this is long-chain fatty acids, mid-chain fatty acids, and short-chain fatty acids from the milk spectrum. The cool thing is that recently there have been a lot of research on the role of short-chain fatty acids on the udder health itself, and it seems, I'm simplifying a lot, but the rumen is producing itself medicines, and these medicines are short-chain fatty acids. And if you have low short-chain fatty acids, you have also increased disease probability. So basically, we created a system that is analyzing real-time the short-chain fatty acids and comparing with some nutritional markers and other health markers. It looks ugly, but this is prototype for now. But to activate it for the vets, we created a system of alert, so that as soon as the farm is entering a red zone or orange zone, it means that nutrition and other health are together wrong at the same moment. So it means that when you go to the farm, instead of just having an approach based on hygiene and monitoring of the animals, you can also think about the nutrition background of the animals. And you see that, again, you have different situations in farms. What is interesting is that farms have recurrent patterns. So you can follow farms that have chronically some issue on that area. The integration of tank data is proactive, so you can also predict the thing. And we have also shown that you have negative covariation between de novo level and other health level, so somatic cell count, in more than 40% of the time in half of the farm of the study. And there was no impact of the season. As a summary, so cows are heavily connected, so they are constantly giving away data. If we are able to analyze it real time, we can do what we call in RUMExpert data-assisted management or medicine. This is giving away feedbacks live using data. 63% of the cows can be eligible to selective dry cow therapy. The dry cow therapy cure rates can explain 43% of the losses. Nutrition is linked to other health 39% of the time. And these data-assisted management era could result into reducing up to 20% of the food waste of Belgium, considering only the milk. Thanks a lot.
Thank you, Leonard, for the nice presentation of all these new monitoring tools. So, are there any questions in the audience? Or online? No question? Yeah.
Thank you, very nice presentation. I was just wondering, how labor intensive is it to collect the data? Is that very automated, or
it's passive? So it's completely passive. So basically the farmer is the center of the consent. So as soon as the farmer has given away the consent, then the advisor can access to all the data sources based on database. So we are working with Exchange files, but now we are moving to IoT, for example, connections also to the database. So it means that in the future there will be more consent, more complex data to analyze, but we have the chance in Wallonie, for example, to have a nice collaboration with Arcia, Awe, Comité du Lait for a long time. And these people have made possible that the interface, as soon as we have the consent of the farmer, per type of data, so this is quite labor intensive, but basically in the interface it's clicking on a button and the farmer has to click yes, that's it. And now we are, I would say, the first private company connected to WallySmart. So this is we are also experimenting the future of the exchange of data. Wow.
Is there any other question? No, maybe one question. When do you plan to implement these new tools in the field?
So they are already in the field. So they are sold. So now we are selling, I would say, we have 65 users and 270 farms in the platform. So it's still quite small, but we can already see some regional tendencies. So I think that in the future, the more user we will have some guidance at local level, but it's possible also to see health at a more global level. So when you do herd medicine, basically you're looking at a group of cow, but looking at a group of herds is exactly the same as looking as a group of vets, or maybe as a group of regions. So for me, as a veterinarian, I would say that herd medicine can be applied at any level. So in the future, there will be even more insights on these approaches.
Okay, thank you. Thanks a lot, Leonard, for the presentation. And so we can move on to the last speaker of this session. The last speaker of this session is Dr. Tommy van Limburgen. Tommy is a vet and is a managing director at Anitom, a company that provides tailor-made consultancy and support research and innovation in livestock. So Dr. van Limburgen will talk about the Neogiant extract-based formulation in poultry. So please, Tommy, the floor is yours.
Thank you. So thank you also for giving us the opportunity to present our work at this nice occasion. So I will try to, how can I switch these slides? The green one, yeah, thank you, yeah. So I will present some of the work that we have performed in the light of a European project, the Neogiant project. I will first start by giving some more information about the project itself. Now it's a multi-species project, not only involving poultry, but also cattle, aquaculture, and pigs. So the basis of everything that we are using within the Neogiant project is basically a leftover project of the winemaking industry in Spain. And it's the white grape mark, that's a leftover product that currently is being used as a feed for cattle very locally. But by testing these products, it was found that it has a very unique composition of polyphenols, very unique characterization, and that this group of polyphenols have quite some interesting effects to be used in livestock production. So this research was granted a European grant, and we have now 3 large parts using this product as a feed additive in feed or in water additives for poultry, pigs, calves, and in fish. We are also looking at therapeutic use of this project, not only using it, for example, for prevention of enterical disease, but also for specific diseases where we are looking, for example, gram-positive mastitis issues. We are looking to see Staphylococcus hiecus, mainly the gram-positive range of bacteria, but there are also some positive effects on gram-negative bacteria. And then a third part is where we are looking to see if this product or one of the formulations of the product can be used to replace the use of antibiotics in semen extenders, mainly in pigs, but also in some other animal species. So as Anitom, we are one of the industrial partners, and we are work package leader for all the animal trials involved in this project. So the majority already said that we are trying to use a local biomass, which is now used as a feed, and we are trying to bring it to another level in order to create a sustainable market for it, or to solve some of the issues using a non-antibiotic approach in this case. So the aim is not to use it as a replacement for antibiotics, the primary aim, but we want this product in certain areas of livestock production to find its place and to reduce the need for antibiotics to be used in livestock. The project also has one non-European country, being Argentina, so we are still looking forward of going over there, but till thus far it hasn't happened. Then one slide to show our company Anitom. So we are located in Belgium, 1 hour from Brussels, to the east side in the beautiful region of Haspengouw, where we have our facilities where we run our poultry trials. So it's basically what we are involved in is not only this type of project using feed trials, feed additives, water additives, but we are also a bit broader, looking for alternative protein sources, more sustainable animal production, animal welfare in this case. So this is our task, or our work that I will present today from NeoGiant is basically the work that was performed in poultry to establish. We had a few formulations which were results from earlier work packages to be used inside the feeds. So we did a dose finding study, also a safety study, as a preparation for an EFSA registration, also to see if some modification was necessary on the formulations or that could be used as such. And then also to see if there are potential zootechnical advantages. So the first type of trials we performed were basically a number of treatments, a number of replicates. We use male broilers to reduce the variation of the animal because there is quite a high variation or a large difference between female and male broilers in the weight gain and their production. So here you can see we had 3 types of dosage, the control group 750, 1500 ppm, which is very high. And then we had 2 groups which were challenged with the coccidiosis infection, which is a unicellular parasite in not only poultry, but in this case in broilers, causing a lot of antimicrobial usage in the field. And then we had some samplings and some moments that there were activities during the trial. So one of the important things, of course, is when you do a feed trial that you test that your active compound is present in the feed. So it starts over there. But we showed this slide to show that the product as such has a unique polyphenolic fingerprint. And this is also what we wanted to find in our feed, because feed and rough materials all contain polyphenols, but it's the combination of a certain group and certain groups, in this case mainly the catechin and the epicatechin, which can cause the beneficial effects in the field. We also did drinking water trials. So in this case we used a high number of male broilers and 2 types of formulation. Apart from this, the animals were intensively monitored during the trial, but also quite some samples were taken to test not only the health parameters that could be seen live animals or scored during necropsies, but also to see if there are, in terms of the immunological parameters, for example, if we can see that there is an advantage or a disadvantage in certain products or in certain areas. Also to see if we see differences in the microbiological content of certain areas in the gut. That's why we focus not only on the small intestine, but in poultry also the chica are quite important. This one I will rapidly show. So these are one of the first trials we did in the with the feed additive, where we had only a limited number of parameters which were showed some effect. I will start to explain the slide first, perhaps. So in broilers, you have, you have certain stages or periods of a certain feed that is provided to the animals. We call it the starter period, a grower period, and a finisher period. And then in the end, you have the overall period from day 0 until day 35. These are conventional broilers. So in general, in field conditions, the majority of the antimicrobial usage and the majority of the stress on the intestine is during the grower period, by the end of the grower period, the beginning of the finishing period. This is where a lot of things happen in the animal, not only from an immunological point of view, but also feed changes. Certain parasites or diseases can occur there. So we expect the most of the results, or the most of the effects of the polyphenolic compounds at times when there is stress in the animal. So our target is mainly there. So we see some borderline significant results. In the feed, we were not very happy. It was only a limited results. On zootechnical parameters, from the immunological parameters and the microbiological parameters, which are still ongoing to be analyzed, they were more promising results. But in case of a drinking water application, at a much lower dosage, because now we are only looking for 2 formulations of the product at the 100 ppm and the 100 ppm range, we saw that there were interesting results in the grower phase, showing that when the animals have more stress, that the product could have its effect over there. So we are now, these are 2 compounds which are now being reformulated because there were some practical aspects regarding to the application that need to be improved. And afterwards we will redo these tests to further investigate the results. So apart from the zootechnical work that is being done, also in later aspects to market the product when it is finished, and for the EFSA registration. We also look for other aspects related to health in general. And one of the tests that you can do is, for example, looking for how it can reduce oxidative stress in the animal. So we look, in this case, it's a test called T-bars. It measures lipid peroxidation. So basically, the lower the value, the better. So the lower the value means that there is lower peroxidation, means that there is lesser oxidative stress, and that it's a better gut health in this case. Or in this case, sorry for the breast muscle. But it's an indication of the antioxidant or the oxidative stress of the animal at that time. So we see with increasing dosages, treatment 1, 2, 3, we're increasing dosages, that the T-bar value is going down. But more importantly, if you look at group 4 and 5, group 4 was orally challenged with coccidiosis. So they had parasitic disease. And the group number 5 was not, both 4 and 5 were challenged with coccidiosis. Group 4 was the control group, and group 5 received the highest dose of the polyphenolic compound. In showing that it can reduce the oxidative stress until levels of a non-infected bird. So which makes it quite interesting, or gives it quite a potential to be used in broilers later on. This is the summary, a bit of the conclusions I already mentioned. So we see most of the effects in the drinking water trial or in the drinking water application during the grower phase, which is logical because we expect most of the stress occurring at that time. We see, which was not shown, but that from histological point of view, that the gut also looks more healthy in the groups that have received the higher dosages. The T-bars I just mentioned, that they were significantly lower at in the group that was challenged with coccidiosis and received the high dosages. And for the other analysis, we are still waiting for the results, but they seem quite promising. So I hope in future symposia, or perhaps next year in this one, that we are able to present more information about this products and also the other animal species. I would like to invite you to check the website of NeoGiant, where you will find also a lot of information of the other animal species and the most recent results. This is result of a group, of course, so I want to thank all of the people involved for their part of the work. Thank you very much.
Thank you, Tommy, for the presentation. So we have time for question. I don't know. Online, maybe? Oh.
Thank you, Etienne Thierry, University of Liège. Thank you for this interesting speech, because we need, of course, to have robust data in order to demonstrate the efficacy of these polyphenol feed additives. My question would be more, maybe very basic and more on the principle. Usually, we assume that red fruits are more concentrated in polyphenols. My question is very basic. Why do you use white grape extracts instead of red grapes?
It's not. It's a very good question. So red grapes are known, or red fruits are known for their polyphenolic content. But in this case, the white grape, it's more specifically about the footprint or the fingerprint of the combination of these polyphenols. So it's the combination of mainly the catechin and the epicatechin and the others that are shown in the slide that are thought to be responsible for the effects that we see. So, but it's a good question, and polyphenols are not only there in fruits, but in many components. So it's continuously, when doing analysis, checking the backgrounds of polyphenols naturally occurring and what are we adding, and how can we see the effects on that. But it's correct, and there are already some products on the market based on red grapes also.
Thank you for your presentation, Bob Roesmans again. You talk about poultry, pigs, calves and fish. Have you thought about maybe in the future using it for dogs? Even though we are all aware that grapes can be toxic, of course not because of the polyphenol, but because of other agents. But could you just try to extract the one that is interesting? And then I'm definitely thinking about the usage against coccidiosis, which can be a problem with puppies.
Yeah, we so in this project, which was part of the Green Deal, we are mainly looking for livestock and improvement in that direction. But it is correct that the pet industry is also very interesting to see where there are potential effects. We are not looking directly for Emeria or coccidiosis applications, but we have one side stream of research looking for a topical application for ear treatments, for otitis. Yeah. So because it's not only an antibacterial, but also there could be some effects to the yeasts, etc. So, but that's still ongoing, so I cannot elaborate much more on that.
Can you give a bit more details on the drinking water application? There's already in poultry quite a lot of treatments that are given in the water. Is it a continuous intake, and is it also, can we combine it with the other products that are given?
Yes, so it was in field conditions. So we, in the first set of trials, the first 7 days, the treatment was not performed. So because we are running the farm antibiotic free, basically. So we are focusing on water disinfection, etc., etc., etc. So we don't want to change this protocol because it works for 2 years and we don't want to adapt it. So after the age of 10 days, the starter period is finished, we rearrange the pens that we have the start of the trial. And then from 10 days of age until 35 days of age, the product was provided to see where we could see the best effects, because at that stage we didn't know where we would expect most of the results. So we only stopped it for a drinking water vaccination, 24 hours, and then the product was given. But in future, given the fact that if you want to, to answer shortly, indeed there are many products on the market aiming for the same type of treatment or the same type of solutions. I think it will in the end find its way between all of that. But at this stage, there is no reason to think that it could influence negatively or positively with products which are still on the market. But it should be investigated more into detail, because if you are looking for mineral additions, for example, then I would I would advise to keep it completely separated. But if you are looking for other phytopharmaceuticals, it will probably depend on the solvent which is in there and the reactions involved. But those things, it's still too early. We are not aiming for this type of test today, but it's a correct remark.
Thank you.
A last question?
Yes, I have one question from Vibeke Jensen. How is the palatability in drinking water?
Very good. I don't know where you are. But
it's a question from home.
It's an online question.
Oh, sorry.
Yeah.
Yes, so no, it's very good, because we kept also records, of course, of the drinking water consumption, and we saw that it was slightly higher in those groups which were treated in comparison with the control groups. So it was not significantly different, but it was slightly higher.
Okay, thank you very much, Tommy, for the presentation and the discussion.
Thank you.
So we are already at the end of this symposium, so it's my pleasure to close this symposium, but don't worry, we will meet again at 1 p.m. for the afternoon session on the antimicrobial resistance communication in Belgium. So I would like, on the name of all the organizing committee, thanks and warmly thanks our invited speaker, Jean-Yves Madec from ANSES, and all the speakers of this morning. It was really nice to hear all the presentation. I would like also to thank all the authors of the posters, and I remind you that the posters are present outside of the room, so take a look to the poster, and they are also online, so you can also follow the poster session online. The video with the recording of this symposium will be available soon also on the AMCRA website. And I would like also to thanks on the name of all the organizers, the organizing committee, the AMCRA, the Federal Public Service Health, Food Chain Safety and Environment, the Federal Agency for the Safety of the Food Chain, the Federal Agency for Medicines and Health Products, and Sciensano to allow the organization of this symposium. So now it's the end, but we will meet around sandwich just outside of the room.